previously healthy 4-month-old boy presents to the fever, chill, cough, runny nose, watery eyes, and he had blood pressure of 115/76. The heart rate is 84, respiratory rate is 14. Physical examination revealed clear lung sounds bilaterally. His mother reports that his brother has been having similar symptoms. A nasal swab is obtained and is diagnosed with influenza. Assuming this is the children's first exposure to influenza virus, which of the following immune mechanism will most likely function to combat the viral? So this patient will have a natural killer cell induced lysis of infected cells. So eosinophilic mediated lysis of infected cell is usually seen in parasites. They release what kind of protein? That is very important. Eosinophil release major basic protein. Virus specific immunoglobulins to remove the free virus. This is usually not seen. The B cell role in the virus fighting ability is usually limited. Complement mediated lysis of the infected cell. The innate immunity does not play important role in the viral particles. Presentation of viral particles on MHC class 2 of CD4 T cells. This is usually seen during second infection. They are asking about the first. Okay, and let's go through the pathophysiology, how they occur. So we have to remember if we have to remember what is the function of natural killer cells, I would just remember these two diagrams that are very important. So let's go through the text first. So what are the functions of natural killer cell? Natural killer cell is a lymphocyte member of innate immune system, and as natural killer cell is more similar to CD8 positive T cell. Both uses the porphyrin and Gran enzyme to induce apoptosis in virally infected cells or tumor cells. So these both are very similar in terms of how they induce apoptosis. One of them is porphyrin, where they puncture the hole into the cell membrane and then they release the Gran enzyme, the type of enzyme that induces the apoptosis. Okay. Activities are enhanced by IL-2, IL-12, interferon gamma, and interferon beta. If you have to remember one of the ILs that is linked to natural killer cells, I would suggest remember IL-12. Natural killer cells also are activated by interferon gamma, but most commonly it's IL-12 that increases the natural killer cells activity. And this both are the mechanisms how they are activated. The first one we'll discuss in this image. It is induced to kill when there is exposed to non-specific activation signals on target cell or the absence of inhibitory signals such as MSC class 1 on target cell. So here they are trying to say is whenever we want to stimulate natural killer cells, we have to kill, there should be the absence of inhibitory signals such as MSC class 1. So remember, MHC class 1 are exposed on all of the nucleated cells of the human body. So whenever there is no MHC class 1 on the surface, it's usually seen when there is virally infected particles. You can see virals inhibit the class MHC expression. So there is no MHC class 1. So there is an absence of inhibitory signals. One is activation signals and one is inhibitory signals. Whenever there is activation signals, then natural killer cells will start engaging and it will kill the cell. But whenever there is inhibitory signals, it will rely on inhibitory signals and it won't kill the cell. You can see here there is an activation receptor, that is activation ligand for natural killer cell, and it recognizes the MHC class 1, and that is a inhibitory receptor. So there is a inhibitory signals and there are also activation, and the natural killer cells are not activated and there is no cellular killing. But in virally infected particles, you can see there is no MHC class 1 and there is no inhibitory. So natural killer cells will activate and cause this infected particles to die. What is the other type of mechanism how they are activated? They are also killed via antibody dependent cell mediated cytotoxicity. We can remember ADCC, antibody dependent cell mediated cytotoxicity. In this, there is a CD16 that binds to the Fc portion of IgG and activating natural killer cells. Remember, this is antibody and this is a FAB portion, FAB portion, and this is Fc portion. So CD16 that will bind to this part of the, and this causes on the IgG, CD16 will bind to the IgG's Fc portion and causes activation of natural killer cells. Okay. You can see in this diagram it's illustrated. So in the surface antigen, whenever any kind of infections occur in this cell, they digest that certain parts of the infected virus and they present on the cell surface. This cell surface antigen is recognized by antibody. It's usually IgG, and this IgG is linked antibody that are recognized by natural killer cells. You can see, and it usually recognized by CD16. So natural killer cells have two types of CD on their cell surface. We have to remember is CD16 and CD56 positive. So this CD16 surface marker on the natural killer cells will recognize the Fc portion of IgG and causes antibody mediated killing via natural killer cells. So this is a function of natural killer cells. Okay.
Thanks for watching. Please give a thumbs up and subscribe for more.